Well, great. I'd like to thank Francesca and the organizers for the opportunity to speak here today. Although this is not Marinus' first time presenting at this conference, it is my first time here at the meeting. So I'm looking forward to learning and interacting with many of you over the course of the next day and a half. Speaking of when Marinus presented here last, we were communicating some open label phase 2 data of Ganaxolone and PCDH19. I'm excited to be here today to demonstrate the significant progress we've made in this clinical development program over the last year and a half, culminating with the initiation of the first ever phase 3 study in this indication. So today I've titled my talk Biomarker-Stratified phase 3 study evaluating Ganaxolone in children with PCDH19 related epilepsy. Before I get into the clinical development program, in case some of you may not be aware of Marinus, we are a small pharmaceutical company based outside Philadelphia in the United States. We're only about 35 people on the team and with a very deep clinical pipeline centered around Ganaxolone. And again, our core focus is on improving lives of patients with rare epilepsies. The picture on the right was just a few weeks ago. Many members of the team participated in an event and provided funding for the PCDH19 Alliance. Here's the Ganaxolone clinical development pipeline. We have working on 3 different clinical indications in epilepsy. We have a phase 3 program in CDKL5 deficiency disorder. We just read out data on a refractory status epilepticus program. But today I'm here to focus about the program in the middle in our work in PCDH19 related epilepsy and how we have recently initiated our phase 3 program called the Violet study. For those who may not be familiar with Ganaxolone, I just wanted to talk about its basic mechanism in the course of the next 3 slides. Ganaxolone is a synthetic analog of endogenous allopregnanolone. Mechanistically, it's very similar in how it acts on synaptic and extrasynaptic GABA-A receptors. Structurally, it just has an added methyl group. On the picture in the bottom, you'll see circled in white. And what advantages this brings over that of allopregnanolone is it renders the molecule orally bioavailable, and it does not convert back to the metabolite with activity at the progesterone receptor, allowing for chronic dosing. It is a GABAergic drug, so similarly, it aims to enhance inhibitory signaling within the brain. However, it is differentiated from other GABAergic AEDs in that it targets a unique binding site on the GABA-A receptors distinct from that of benzodiazepines. Also, which is different than existing AEDs, particularly benzos, is that, as I mentioned previously, it acts on the synaptic, which benzos act on, but also the extrasynaptic receptors. In some cases of prolonged seizures, the synaptic receptors downregulate. So having a molecule that could target the extrasynaptic receptor could be of added benefit. Ganaxolone has been in clinical development for a couple of decades now, and so what advantage that brings is that we have a well-known and established safety profile on the drug. It has been dosed in over 1,600 children and adults, to be more specific, more than 200 pediatric patients. We have dosed patients, many of them more than 2 years, even one of them have been treated for more than 4 years on Ganaxolone. The most frequently reported treatment emergent adverse events are on-target AEs of a GABAergic drug and are primarily somnolence, dizziness, fatigue, and headache. So I want to transition into the scientific rationale for Ganaxolone in PCDH19 related epilepsy. Around 5 to 7 or 8 years ago now, there was reports that there may be an endogenous allopregnanolone deficiency in those with PCDH19 when compared to age-matched controls. A lot of the great research from scientists and clinicians are those that are in the room here today, and that's what this figure here shows from the seminal paper in 2015. So with Ganaxolone being a synthetic analog of allopregnanolone, this established the scientific rationale or hypothesis for treating these patients with Ganaxolone. So around 4 years ago now, we ran an open-label phase 2 study of ganaxolone in this patient population, and these data were reported previously. Just want to go through them quickly, but then talk to you about what developments have been made over the last year and a half. 11 patients were enrolled at 6 clinical sites, most of them in the US, 1 site here in Italy. The primary efficacy endpoint was the % change in seizure frequency per 28 days relative to the baseline. The graph there shows all 11 patients and their response to ganaxolone as it relates to the primary efficacy endpoint. What you can see in the intent to treat, or ITT population, there was a modest 25% reduction in seizure frequency when compared to baseline. What we did retrospectively is that we conducted a non-classical responder analysis, non-classical in that we used the responder definition of having experienced a reduction in seizure frequency of 25% or more. And the reason for this is that it allowed us to more equally bifurcate the population to see if there were any trends between those that responded better to ganaxolone compared to those that did not. Going back to the endogenous neurosteroid hypothesis, we did measure these endogenous neurosteroids in the 11 patients using a highly sensitive gas chromatography mass spectrometry technique. And what we saw was that perhaps as a population, these patients have low levels of these various neurosteroids, but on an individual patient-by-patient basis, that may not in fact be the case. And in fact, those that had these low levels of allopregnanolone sulfate were more prone to respond favorably to ganaxolone when compared to those that had very high levels of allopregnanolone sulfate. Now, I do want to acknowledge that on the previous slide, I talked about allopregnanolone, and here I'm talking about the sulfated form. We are currently trying to explore the mechanism and correlation between the sulfated form and the non-sulfated form. We do have some data that demonstrate that there is a positive correlation between allopregnanolone sulfate and allopregnanolone, with the sulfated form being the predominant analyte in plasma. And the hypothesis being there is that the sulfation occurs primarily in the periphery. So this got us thinking, is there a way we can utilize these data to try to prospectively identify patients that perhaps may be more prone to respond favorably to ganaxolone? And so that's what the dashed line here shows. And I'm sharing the same data that I presented on the previous 2 slides, condensed together onto 1 slide. And so what I'm showing you here, though, is what if we draw this hypothetical stratification boundary? I've drawn it here at 2,500 picograms per mil allopregnanolone sulfate. And I've separated the population into what we're calling biomarker positive, which are those with lower levels of allopregnanolone sulfate, and biomarker negative, those with higher levels of allopregnanolone sulfate. What we see here is that we may be able to identify an enriched patient population in the biomarker positive stratum that perhaps will have an enhanced treatment effect to ganaxolone. And what we see in these 7 patients is a markedly improved seizure reduction rate of 50%. And so we do recognize the limitations of this analysis. It's in 11 patients. It's in a post-hoc analysis. However, nonetheless, we do feel like these data were compelling enough to study further in a larger phase 3 study. So now I want to explain how we leverage those information and utilize them in the design of the phase 3 Violet study. So here's a diagram showing the design of the Violet study, where we propose enrolling all eligible PCDH19 patients and then subsequently stratifying them based on their baseline allopregnanolone sulfate levels. Again, knowing that what I just presented only came in 11 patients only allowed us the ability to come up with a hypothesis. It wasn't definitive enough where we felt like we should exclude patients with high levels of allopregnanolone sulfate. In fact, we want to study this relationship further in a larger clinical study like this. Within each stratum, though, the same randomization scheme will apply, where they'd be randomized 1-to-1 to active ganaxolone or adjunctive ganaxolone or adjunctive placebo. The duration of the study and its components are shown below. The baseline period is 8 to 12 weeks. The difference between 8 to 12 depends on some protocol amendments. And then the study is 17 weeks double-blind. We're planning to have 35 global sites in the US, Europe, and others. There are 4 active sites open in Italy currently. The primary endpoint is the % change in seizure frequency. Again, you can see in the dashed box above, we are proposing to do our primary efficacy analysis in the biomarker positive stratum to test that hypothesis that there could be an enhanced treatment effect there. I do want to acknowledge that, again, sometimes the epilepsy and seizures we recognize are not the most significant clinical manifestation in this patient population, so we also are looking at other behavioral endpoints and sleep-related endpoints as secondary endpoints, and we recognize the importance of that. The key eligibility criteria that I want to highlight here is, again, a pathogenic or likely pathogenic PCDH19 variant. We're looking for female patients. Again, we recognize that the prevalence and the identification of mosaic males is increasing over time. However, we are looking at just solely females based on our experience in phase 2, looking at only females and trying to study the allopregnanolone sulfate hypothesis. And then also having 12 primary seizures defined below over a 12-week period prior to screening. The reason for these primary seizures, we like to think of them as the 3 C's, those that are most common, most consequential, and most easily countable seizure types. Exclusion criteria are previous exposure to ganaxolone. Again, we do recognize that the hallmark phenotype of epilepsy phenotype are these clusters of seizures. However, for this clinical study, we are looking to identify patients that have a more regular seizure pattern, and so thereby excluding patients that have greater than 8 consecutive weeks of 2 months of seizure freedom during a 3-month period would not be eligible for the study. We do hope that if successful in the study and that this drug could provide benefit to all PCDH19 patients, but in terms of running a clinical study, we are looking at a particular subset with perhaps a more regular seizure phenotype. And then the use of non-approved CBD is prohibited. Again, with the approvals coming on board, those will be allowed in the clinical study. I just wanted to spend one slide and just highlight on the importance of what a placebo-controlled pivotal trial, what it allows us to do, and what the reason for doing so is. It's designed to isolate the effect of an active drug, if there is one, and minimize bias when assessing safety and efficacy outcomes. It's also important to know that this is adjunctive placebo and it does not deny the patient care or therapy. They will continue to be maintained on their background regimen of various AEDs. So again, it's no different than receiving standard of care. Patients in this study would be randomized 1-to-1, which means that a patient has a 50% chance of receiving active ganaxolone or a 50% chance of receiving placebo in addition to their background AEDs. Patients that do complete that 17-week double-blind phase will have the opportunity to transition into the open-label phase where all patients will receive ganaxolone. So in conclusions, again, we feel that there's a strong scientific and mechanistic alignment for ganaxolone in this patient population. We feel that the preliminary data that we show here suggests that allopregnanolone sulfate may be a predictive biomarker for ganaxolone in this indication, and the phase 3 study is designed to demonstrate if that is or is not in fact the case. Again, it just allowed us, the phase 2 study allowed us to establish the hypothesis that this could be worth exploring, which we have done so or are doing so now in the phase 3 study. And as I mentioned, we are currently enrolling with many sites open in the United States, and like I said, 4 sites open in Italy with some of the investigators here in the room today. Again, please feel free to contact me if you have any questions, or if their time allows, I'm happy to take them now.